by Q. You have to learn. It's discovered by the scientist. The name of the scientist is Bloomberg. It is the only hepatitis virus which is DNA. Rest all are RNA. It is the only hepatitis virus which is double-stranded. Rest all are single-stranded. Okay, it belongs to the family Hepatnaviridae. I told you the families of all 5. Hepatitis B virus belongs to Hepatna. A belongs to Picorna or Enterovirus, and B belongs to Hepatna. Please learn that. Okay. There are 3 morphological forms in hepatitis B virus. How many forms are there? There are 3 forms. There can be spherical form, tubular form, and compact form. The compact form is ultra important, which is known as Jane particle. Jane is the name of the scientist who discovered it. See, this is spherical, this is tubular, this is Jane. This is spherical form, this is tubular form, looking like a tube. The spherical is sphere, and the Jane. Jane is compact. The compact is Jane. So antigens are different. Spherical form have only one antigen, hepatitis S antigen. Tubular also have only one antigen, hepatitis S antigen. Can you see this one? So this is hepatitis S antigen. They have only one antigen. They also have one antigen. Okay. But what about the Dane particle? It is having hepatitis S. But along with hepatitis S, it is having 2 more. Hepatitis C and E. See the diagram of the Dane particle. You can see this is spherical form here also, having only hepatitis S. This is tubular, having only hepatitis S. So hepatitis S is present. But in Dane particle, hepatitis S is there. Along with that, hepatitis HBE. This is S, this is E, and this is C. So total 3 antigens are there. C for core. E is pre-core. E is pre-core. Okay? And S is surface. 3 antigens are there. Shall I draw it for you? I will draw. So see, this is the virus. This is the genome of the virus containing double-stranded DNA. Okay. Now on the surface, the antigen which is present, this red color I am drawing. This is hepatitis S antigen. Okay. This is hepatitis HBS antigen. This is known as HBS antigen. HBS. Now there are 2 more antigens present. Here I am drawing the pre-core. This is pre-core. And one I will draw the core. Below that there is core. So 2 more antigens are there. Can you name them? The pre-core is hepatitis E antigen. And the core one is known as hepatitis C for core. The innermost. C antigen. So C for core. And for surface, that we can understand. S is outermost and C is innermost. The between the 2 is E, that is pre-core. So there are 3 antigens are there. And what about the 2 more morphological forms, spherical form and tubular form? The spherical and tubular form have only hepatitis S antigen. They don't have C, they don't have E. They have only S. You can see, I am drawing. They have only hepatitis S antigen. Okay. So these are the 3 morphological forms I told you. What are the 3 morphological forms? Spherical, tubular. I am interested in Dane. So Dane. I am interested in Dane, the 3 antigens. The 3 antigens of the Dane. Okay. Now coming on the viral genome. In the viral genome, there are 4 genes present. I am talking about hepatitis B virus, which is a DNA virus. 4 genes are present in the genome. Learn the name of the genes. This is a double-stranded DNA having genome. Okay. So S gene, C gene, X gene and T gene. Let's talk about S gene. S gene. S gene have pre-S1 and S2. Can you see? This is pre-S1, this is S2 and this is S. So this is S gene. The different portions of the S gene. They give rise to hepatitis S antigen. These genes give rise to hepatitis S antigen. What is the hepatitis S antigen? It's a protein. All the proteins are coded or they are formed from genes. So hepatitis S antigen का gene कौन सा है? The gene is S1, S2 and S, which are formed by the different portions of the S gene. The second is the C gene. C gene have pre-C and C. You can see this is pre-core and this is core. Pre-core and core. They give rise to hepatitis E antigen as well as hepatitis C antigen. So E and C, they both are formed from C gene. So this is formed from S gene and these are formed from C gene. And out of the 4 genes, 2 are present. So S give rise to hepatitis S antigen and C give rise to hepatitis E antigen and hepatitis C antigen. I hope you all got it till now. Now what about X and P? Coming on the third gene. The third gene is the X gene. X gene code for an antigen, hepatitis X antigen. It is not formed normally. It is formed during carcinogenesis when hepatocellular carcinoma. During carcinogenesis, it is formed. It is a carcinogenic gene. X gene, it is activated during carcinogenesis. And P gene code for three enzymes: DNA polymerase, reverse transcriptase, and RNA. For these three. So can you say the four genes? What are the four genes? S gene, T gene, X gene, and P gene. What does they code? These are the different types of genes present in the genome of hepatitis B virus. Genes of hepatitis B virus. So can you tell me what does they code? S gene code for what? Coding an antigen. The name of the antigen is hepatitis S antigen. T gene also codes for two antigens. Hepatitis E antigen and C for C. Hepatitis C antigen. So all the three. Total we have three antigens. All three are done. All three are done. Now coming on X. X code for hepatitis X antigen, which is formed during hepatocellular carcinoma. So it indicates carcinogenesis. It is not normal. It's abnormal. And T codes for three enzymes. What are the three enzymes? DNA polymerase, RNA transcriptase, reverse transcriptase and RNA. This is reverse transcriptase. So learn the name here. So that is a summary of the genome. Now I am coming on the mutants. How many mutants are present in hepatitis B virus? In hepatitis B virus, how many mutants are present? There are three types of mutants: pre-code mutants, escape mutants, and YMDD mutants. Let me explain you one by one. So pre-code, they have mutation in one of the gene. I told you four genes, no? S gene, T gene, P gene, and X gene. So which gene have the mutation and having these mutants? So let's talk about first mutant, pre-code mutant. In the pre-code mutant, C gene ka that portion get mutated which form HBE. So HBE is not produced. HBC is produced, S is produced, but HBE is not produced. So there is no production of HBE. That's why it is diagnosed very late. That's why there are high chances it will convert into severe chronic hepatitis and it will lead to cirrhosis. So pre-core mutant. In pre-core mutant, the C gene is mutated. So HBE antigen is not formed. Since it is not formed, the chances of chronic hepatitis is high. Cirrhosis is high. And it is diagnosed very late. So this is the consequences of mutant. The second type of the mutant is escape mutant. Here S gene is mutated. What does S gene form? Hepatitis S antigen is not formed. Since hepatitis S antigen is not formed, it cannot be diagnosed. Again, late diagnosis. And are you vaccinated for hepatitis B? I guess everyone, all the healthcare providers, doctors, nurses should be vaccinated. If you are not vaccinated, get it vaccinated as soon as possible. So I am vaccinated for hepatitis B. So in vaccine, the inactivated hepatitis S antigen is given. So our body forms anti-HBs antibodies, which give protection for the future. But suppose someone is vaccinated and someone is having this escape mutant version of the hepatitis. So hepatitis will occur, but HBs antigen will not be there. So vaccine ki antibodies cannot neutralize it. So it cannot be prevented by vaccine. Vaccine can prevent all type of hepatitis B virus except escape mutants. Escape mutants cannot be prevented by vaccine because in vaccine the antibody is anti-HBs antibody and in escape mutants HBs antigen is absent. So what does this antibody will neutralize? They cannot neutralize, no? Antigen hi nahi hai, so how they will neutralize? So that is the escape mutation. The third is YMDD mutations. Now the person already have hepatitis B virus. They are on treatment. What is the treatment? In the treatment we give one of the antiviral drug, that is lamivudine. Patient is already on lamivudine therapy. They will have a mutation in YMDD locus. YMDD locus present in the virus and they develop resistance for the drug. So these are the three type of the mutants. I told you 4 type of genes and I told you 3 type of mutants. Can we revise it quickly? What are the 4 types of genes I told you? Can you please tell me? The 4 types of the genes and what are the 3 type of the mutants I told you? For hepatitis B virus we are studying. So the genes are, what are the genes? S gene, C gene, P gene and X gene. In the mutants I told you 3 variants. What are the 3 variants? What are the 3 variants I told you here? What are the three variants we have seen here? So the three variants are pre-core mutants, escape mutants, and YMB. I will write on next page. Mutants of hepatitis B virus. Okay. What are the mutants? Let me explain all three. The first one is pre-core mutant. The second one is escape mutant, and third is YMDD mutant. So what is, name the gene which is mutated? Here the gene which is mutated, the name of the gene is C gene. Here the name of the gene which is mutated is S gene. Here YMDD locus is mutated. What is the consequence? Because the C gene is mutated, HBE is not formed. This antigen is not formed. HBC can form. In the C also we have two portions. The portion which is coding for HBE is mutated. So HBE is not formed. S form HBS antigen. Here HBS is not formed. And YMDD locus is mutated. So the patient is on lamivudine therapy. They will develop the resistance. Now tell me the consequences. Here because HBE is absent, it is diagnosed late. Chronicity is more, cirrhosis is more. Here again they are diagnosed very late, or rather they are undiagnosed at all. And here the vaccine cannot prevent it. That's why they are known as escape. They can escape the vaccine also. That is, the vaccine cannot prevent them. And here, resistance on lamivudine. This is the consequences of the three mutants. Please learn the properties of the three types of the mutants. So we are done with the three properties also. Now I am coming on the replication of hepatitis B virus. How does hepatitis B virus replicate inside the host cell, inside the hepatocyte? So first thing, I told you, hepatitis B virus is the only virus which is having DNA. The double-stranded DNA. Rest all have the RNA. This is the only virus which is having DNA, rest all have RNA. Okay. So here the partially double-stranded DNA first converted to fully double-stranded DNA. Initially they have partially double-stranded DNA which is converted to fully double-stranded DNA by an enzyme polymerase. So this is taking place by polymerase. Now this fully double-stranded DNA will produce two types of mRNA. One is simple mRNA and one is pre-genomic mRNA. Pre-genomic RNA. This is transcription. The pre-genomic RNA will produce the progeny DNA, the daughter DNA. It is known as transcription, reverse transcription. From RNA to DNA is formed. And mRNA is forming the protein, the viral protein, the progeny protein. This is known as translation. mRNA to DNA, mRNA to protein. They will combine together and daughter virion cells. I will say it again. So I am having a virus, hepatitis B virus. This hepatitis B virus is having partial double-stranded DNA. First I will convert the partial double-stranded DNA to complete double-stranded DNA. For this an enzyme is required. The name of the enzyme is polymerase. Now after that, this double-stranded DNA, it will form two types of RNA. One is pre-genomic RNA, it is an RNA, and one is messenger RNA, mRNA. Okay? So DNA to RNA conversion. What is it known as? DNA से बना RNA. It is known as transcription. DNA to RNA. Okay? Now this pre-genomic RNA, this will convert into DNA again. It will convert into DNA. The daughter, the virion DNA, the daughter virion DNA. And here the mRNA get converted to protein. Got my point? So what I am saying here, RNA to DNA, it is known as reverse transcription. And mRNA to protein, this is known as translation. So here you can see first transcription is taking place. You can see here, just a second. First transcription is taking place, then reverse transcription taking place, then translation taking place. Finally, the two things combined, the DNA and the protein and the daughter cells, the daughter virions are formed, the daughter virus is formed. Got it? If you got it, it's good. If you didn't got it, just learn the important points. You will not get question from that. Just for your understanding, I told you. So you can see this is the host cell. Inside the host cell, you can see this is the virus entering. After entering, you can see first partial DNA converted to complete DNA. After that, two type of RNAs form, messenger RNA and genomic RNA, and they are forming the protein and the genome, and finally it is combining and daughter cells are formed. Now coming on mode of transmission, there are 4 modes of transmission for hepatitis B virus. What are the four modes of transmission for hepatitis B virus? Let me explain you the four modes of transmission. Parenteral, sexual, perinatal, that is also known as vertical, and rarely there is direct skin contact. For hepatitis A virus, it was fecal, don't forget. And here we don't have fecal. So parenteral route is via blood products or via needle prick, common needles, common syringes. So that is the most common route in developing country. The most common mode is through needle prick or transfusion. It is here HPV virus is more infectious than HIV and HPV. As small as 0.00001 ml of the blood can transmit the infection. Just suppose here we can see a needle. Can you see a needle here? So this needle is used in the patient having hepatitis B. So needle contains the blood. Blood is stick on the needle, and the same needle is pricked by mistake to some other healthy person. So 0.00001 ml of the blood is transmitted here. It can get the infection. It is as small dose as that. So that is parenteral route. The second route is the sexual route. It's rare, but it's possible. It's possible. Homosexual males are at high risk. Homosexual males. The vertical route is the perinatal route. I told you, from mother to fetus via placenta. Via placenta, from pregnant lady to fetus, it can transmit. It can. The transmission can occur during uterus also, during delivery also, and after delivery during breastfeeding also. Maximum chances during delivery. That is vertical. And rarely direct skin contact is also possible. So 4 routes are there. I told you 4 routes. Now coming on the epidemiology, based on the epidemiology, we divide the countries into 3 categories based on the carrier rate. If the carriers are less than 2%, such countries are known as low endemicity. If the carrier rate is 2 to 8%, such countries are known as intermediate endemicity. And if the carrier rate is more than 8%, such countries are known as high endemicity. So high endemicity is there. India will come in intermediate endemicity. Okay, that is the epidemiology. World Hepatitis Day is celebrated every year, 28th July. And the theme which was observed in year 2020, it was hepatitis free future. Okay, so that is the thing. Now coming on the clinical features. Incubation period, I told you the average is 60 to 90 days. For B also, for D also, I already told you 60 to 90 days. There can be subclinical infection, acute infection, chronic infection. If it is acute infection, again like hepatitis A, pre-icterus phase in which nausea, vomiting, and icterus phase the patient has jaundice, yellowishness of the skin and the sweat. This is acute hepatitis. Now see this algorithm. You can understand this algorithm very well. If the patients have infection, hepatitis B infection by any route, most commonly parenteral route, sexual route, vertical route, then after the incubation period, patient have acute hepatitis. Acute hepatitis rarely 1% converted to fulminant. I told you all acute can convert into fulminant. Here 1% is converting to fulminant. 1%. The remaining 99%, out of the remaining 99, 95 will recover. Out of the 95 will recover. 5% will convert into chronic. And rarely less than 5 convert into carrier. So recovery, chronic, carrier after fulminant. If there is this chronic hepatitis, there can be 2 types of chronic. Chronic active, chronic inactive. If it is chronic inactive, it will convert into hepatocellular carcinoma and cirrhosis. So I will draw it. Someone is having hepatitis B virus infection. The person will always have acute hepatitis after incubation period. Acute hepatitis. Now acute hepatitis have 4 phases out of the 100% I am talking. Okay, 1, 2, 3, 4. 1% or less than 1% have fulminant hepatitis. Let me write fulminant hepatitis. 90 to 95%. 90 to 95% have recovery. Subclinical or recovery. Okay? Less than 5, nearly 5 or less than 5% have chronic hepatitis that will give the trouble. And less than 5% have carrier. Okay? So carrier will remain as carriers. That will decide the endemicity of the country. Fulminant will die. The fulminant one will die. They have high mortality. They will decide the carrier. They are asymptomatic. Recovery to recovery ho gayi. They are recovered. Now the chronic will continue. They can be active chronic and they can be inactive chronic. If they are inactive chronic now, so they will convert into cirrhosis in future and they will convert to hepatocellular carcinoma. So some percentage will convert into cirrhosis and hepatocellular carcinoma. You can see. So this is the algorithm you have to learn. Now I'm coming on lab diagnosis. In the lab diagnosis, the serological markers. The antigenic markers, they are 3 in number. The antibody markers, they are also 3 in number. Total